So today's topic is multi-omics data analysis, and I'm Ann Holmberg from Qlucore. I will share the slides with you after the presentation, and if you have any questions, just use the chat and I'll try to answer them as we go along, and we will also have a Q&A at the end. Okay, I got one message about bad sound, but I think just... Okay, so let's get started. So I will just switch screen here, so I will stop my video like that. So the background for this webinar and multi-omics is that the multi-omics approaches to data analysis is becoming more and more popular, providing a better understanding of the system under study. So it's common then to work with, for example, combinations of transcriptomic, proteomic, epigenetic, and metabolomic data, and so on. And in this webinar, we will combine transcriptomics and proteomics data and demonstrate how you can quickly identify correlations between genes and proteins analyzed on different platforms. So the general principles is that you, in this case, we have started with two data sets, transcriptomics and proteomics data for the same samples. And then we have merged these two data sets into one, and also included a column with information what the type of the variable is, so gene or protein. And then we have imported this merged data. So let's have a look at how that works. And then we will use correlation to find correlated genes and proteins using Pearson correlation. Oh, let's start by just having a look at the data set so that you understand what we have done here. So here it is, saved as a TXT file. And here you can see that we have 72 samples with different treatments. And then we have a number of variables. So we start with our genes. And when we come further down here, you will see that then we have protein data that we just added here to the same matrix. And you can see that these values are a bit different. Here, for instance, we have very many negative values. So these were probably zeros or very close to zeros before they were log transformed. And that's very common in a proteomic data set that you have zero values, of course. Okay, so a mixture of genes and proteins from different platforms and probably normalized in different ways or so, but when we're working with correlation, we avoid that problem with the different normalizations. So let's come here now. And now we want to import this data set. So we do File, Open with Wizard, find the data set, and then just step through the wizard. Answer the questions as usual. Is this a raw count matrix? No, it's not. The separator is tab. The variables are, the rows are variables. And then we identify the data matrix. So first select the top left cell of the data matrix. Next. And we select the top right cell of the data matrix over here. Variable annotation header cells, here we have those. And then the sample annotation header cells like this. Here we have the data set. So we have 72 samples and 378 variables. And here we are looking at the different treatments. So we have one control treatment and then 3 different drugs like that. And the data set, we have actually here selected our best genes and proteins, the most interesting ones that are really good at separating the groups, as you can see. Now we will switch to a variable PCA plot. So now we are actually looking at the different variables, the combination of genes and proteins here, but we would like to know which are which. So in order to do that, we will use in the variables tab, there is a search tool. So that normally comes up like this. And here I will search the annotation type for gene. So there are 164 genes in here, and they are now here in the search. So by making a copy of search, I now have a list of genes. I do the same for protein, and I find 214 proteins. I make a copy of that, like that. And now I can use these lists. So now I color by this, by just ticking this second box here, and that color. So you can see that we have a mix of genes and proteins. The genes are yellow and the proteins are pink. Now I want to find correlation, and initially I want to find correlation for one specific gene that I'm interested in. So CD74, can close this for the moment. So I have ordered them in alphabetical order here, so I just go and get that gene. So here it is. And by using the mark tool here, I now mark that gene in the plot. So let's move it a little bit so it's easier to see. Here it is. And to be sure it's the right one, I can also use the label tool. So here we are. And I would now like to find correlated variables, both genes as well as proteins, with this CD74 gene. So then we switch to the correlation tool here, like that. And that opens up this little dialog. So first I need to mark my gene of interest here by clicking on it, like that. And now you see there's a little cross in there. And then I just drag the correlation slider to the correlation cutoff that I want. So 70% correlation using the Pearson correlation. And you see that that finds a lot of correlated variables here. I can also look for the negative correlation where I have an opposite expression. And now I want to create lists of my findings, but there are a lot of other variables here that are not within the correlation ones. So then I can use the variance filter in the statistical dialog here, like that. So by filtering away everything else like this, that then reveals the correlated variables like that. And now I would like to save lists of these. So then I create a new list here. I call that positively correlated correlation for CD74, like that. And then I use the list tool and just circle these like that. So now I have them here. And I can add more information here. Maybe I would like to see the type, and I would also like to see the actual correlation, and I can sort on any of these columns like that. that. Of course, we have 100% correlation with CD74, who is actually in here. And then I would like to do the same for the negative correlated. So then we call that negative correlation for CD74. And then we use the list tool and circle like that. So now we have our two lists. Great. And now I would like to, of course, I have the gene and protein in here. That's good. Maybe I would like to visualize this. So let's switch now to a Venn diagram. That and so here I add the gene list and the protein list, and there is no overlap here, we know that. And then I also add my two new lists, the positive correlation as well as the negative correlation, like that. Okay, so here we can see that we have positive correlation, 28 positively correlated genes, and we have where are we now? And we have here 40 positively correlated proteins. And in order to make a list of these, I just click here in the intersection, create new variable list. The list tool must be active for this to work. Create new variable list. Okay. So here we have positive correlated genes. And if we do the same over here, we have positively correlated proteins, like this. Okay, and this is a new feature, so if you haven't discovered it, I'm now showing it to you, the possibility to create lists like this from the intersections. Okay, so that was the first workflow. So let's go over here again. And I also want to remove the correlation now here. So in order to do that, I just mark the correlation tool and press clear again, like that. And then to bring back everything, I set the variance filter to 0 again. Okay, and here are the steps, everything that I did. Now we are going to do it in a little different way. So here, this time we are going to use the search tool to create a list of the, in this case, proteins that I want to correlate with, and then use that list as input. So let's see how that works. So then we use the search tool again and say that I now would like to search the ID, gene protein, and I would like to look for proteins names that start with SPA6 and then wildcard. And then I find these two, okay? And I have them here, so I want to make a copy of that. So these are my two SPA6s. And now I will instead use this list as input. So by ticking the leftmost box here, it means that you are now only using those variables in that list. And you see, I now only see two pink variables here indicating that they are proteins. And let's label them to check that they are the right ones. Yes, they both start with SPA6 like that. And now I use the correlation tool again, and I can either click or just circle them to do like that. Yeah, so this is where we start, and then we drag the correlation slider like this. Maybe we only want the positive correlation this time, like that. So now I find 2 correlation networks here for each of these proteins, and then I can save lists of that in the same way as I did before. And then we clear that again. And we clear that so that we use all, go back to see all the variables again. Okay, and then we move over here where we are going to look at another type of correlation network. So let's clear the labels like that. And so now instead we are going to use this tool here. So in the method tab over here, we have something called network. And you can work either with connecting to the closest neighbors, but then you will always get neighbors. So if we instead use the correlation slider here and look for maybe 85% or so correlation, you see that we find a couple of interesting networks here like that. And then we can try to grab this into a list again. So it's a bit hard to see. So I will actually double click in the plot and zoom in a bit here. So it's a bit easier to see like that. And then I will try to grab only the ones that are actually connected here. So I create a new list and use the list tool, and I circle as best as I can here. So unfortunately the variance slider doesn't work in this context. Okay, and here I now, so let's again also then color by this list that. And now I will pull up a new synchronized plot and make that a heat map, because I want to view this also in a heat map. So I will hierarchically cluster the heat map like that, and I will color by list. That means that I will now color it by the same colors here that I've chosen. And then we can zoom in. And here we have the cluster. And then you can actually easily, by using the list tool, you can make corrections here in the heat map if you believe that variables should be included or maybe not included here. So if it's included and you click on it again, it will disappear from the list, and the other way around. So here you can actually view it in the heat map, and here you also have the names. So these are 2 different views for the same thing. So this is also quite useful. And a third way or fourth way of looking at correlation is to use the correlation matrix, which will give you a full overview of the correlations between all the variables, all the active variables. So here we can now go to File, Export, here, Variable Covariances Correlations. Call that correlation. Put it on the desktop. And pick the annotation that I would like here, and I would like correlation that. And then we go here. Here it is. So I open it. It's a text file that I open in Excel. And here you see then I have the complete list of correlations between both genes as well as proteins, like that. This is also very useful. So in this case, I took the whole data set. You might also take a subset of your data set, of course. Okay, and now we are going to talk a little bit about how you can work with linear regression. So typically you use it when you have a numerical sample annotation like time, dose, and age to find variables that increase or decrease over the annotation. In this case, we are going to use some of our variables as sample annotations and use linear regression to find statistical significant correlations. So let's see how that works. So we will close down this data set like that. And now we will open up another data set. Let's have a look at it before we import it. So it's this one that we are going to have a look at. So let's see what we've done here. So here we've changed it a little bit so that the sample annotations here are in the same column as the gene and protein names, because we are going to use all the genes as sample annotations instead of as variables. So that's the only change that we have done here. So let's import this data set. Open with Wizard. And I have it here in this here. And again, we step through the wizard, no tab, variables, and then select the top left cell of the data matrix. And now the genes, we will want to treat them as sample annotations. So the variable, the actual measured values, the variable values starts down here on the proteins. So this is the first measured value. Next. And then we go all the way to the right and was column 52, I think, here, like that. So those are this is the measured data. And then we click on variable annotation header cells. And again, we cannot be up here because these are sample annotations. So we go down here. And we don't have a header here, but that doesn't matter. I can just click on the first cell of that for the variables, and I bring in that column as well. And then click on sample annotation header cells. I want that one, that one. I don't want that one, so I skip that. I want that one. And then I go all the way down. I press Shift, click on the last value, like that. And that will bring in all the genes as sample annotations together with the two real sample annotations, like that. And then we press Finish. Okay, it looks very similar to before. So in this case now, let's see. As you see, now we have a long list of sample annotations. Before we only had two, but now we have decided to treat the genes as sample annotations. So maybe I would like to work with this gene. So first, if I color by that, it doesn't really make sense because all the numbers here have different colors. So I want to create a color gradient. And I do that by using this tool here. Color arrange as a gradient. OK. Select the first value in the range and press OK. Before I press here, I want to change the color. So I double click in the color box like that. And then I select it. Select the last value in the range. I go down here. I want this color to be blue, so I change that first. And then I select it and press OK. And now I have a color gradient from yellow to blue. So yellow are low values and blue are higher values. And you see that two of the groups here are actually either have low values or high values, and the other two groups are a bit mixed. Okay. Now I want to do a linear regression here. So I pick linear regression, and I want to use this gene to find other correlated variables or statistically significant variables. So I filter down to a good Q value, a good false discovery rate of 0.01, which is very good. And then I find 139 significant variables. And we have them here. We can save a list of that. So let's have a look at these variables in a heat map. So let's change the view here to a heat map. And again, let's do like that. And we will order hierarchically clustered like this. We will color the heat map on this gene like that. And we will also add color for the treatment like that. You know, you can have several colors in the heat map. And again, I might want to change the color of the treatment. I want that one to be yellow like that. Okay, so here we see that this group here, the drug 2 group, here basically all the samples have a low expression for my gene of interest. And here is the pattern for the other ones. And here for this control group, we have higher expression. So this is a way that you can visualize your result. Another way, let's add Q value and sort on Q value so that we have the most significant variables at the top. Maybe we would like to have a look at our results in a scatter plot. So scatter sample. And then we select our gene of interest, here, like that. We want a numerical scale. And then we select the variables of interest here. So let's pick the 4 best ones. Maybe color them on treatment groups. Maybe change protein name to the top. Some of them are very, very long, as you see. And we can move that little information box a bit if we want to, like that. So this is one way that you can, so here we can, for one variable or sample annotation on the x-axis, we can then select up to 64 variables to show on the y-axis. We might also want to compare the results in a line plot. So let's open a line plot. Here we have active samples on the x-axis, and again we can pick which variables to show here. And we might also want to change the order here so that I want to order by this sample annotation like that. And I might want to order here numerically and increasing instead like that. And I want the that annotation here, and I want to be able to see the numbers, so I like that. Yep. So these are well, some examples on how you can visualize your findings. Like that. And the last thing that I will show you is how you can use the Biomarker workbench to get a quick overview and identify the strongest signals, the most interesting results here. So in the Biomarker workbench, which is this icon here, here we can quickly do a batch analysis. So if I say I want to add everything, then you see that I add all the genes that are now sample annotations, and it's automatically suggested that we do a linear regression because these are numerical results, and I also get the treatment annotation here. And then by just pressing run, all of these tests are performed in one go, and here is the overview results. And you see that there are actually, well, find very many significant variables here for each of the genes actually. So this is a data set with very strong signals. So here you can get an overview. Where do I have the most interesting signals? And here in the detailed report, you can go in and look at the details. You can click on each of them to sort the variables and then make a list out of these. So for each of these, you can do that. And you can configure the view here so that we take away some of the columns here, maybe like that. So we show less. So you can click on each of these and find where you have which are the most interesting variables here. And then you can easily create a list by just marking the ones you want and then create variable list. Yeah, like that. And that list will then show up here, and you can work with that. Good. So that was everything I intended to show you in this webinar. So now I'm open to questions. One comment from my side is that if you were new to the software, this might have been a bit much for you to follow. I don't know, or maybe it was crystal clear. If you feel that you would like to have a more general introduction, you can join the webinar on Tuesday next week, where we look at some typical workflows to identify statistical significant variables using t-tests, f-tests, and so on. So let me see. I think I have a couple of comments here. Let's see. So I have a specific question here. Okay, so somebody would like to have a look at the file again. We can do that. And I got a question: is it possible to build a classifier based on proteins and genes? From a technical point of view, yes, because we just view everything as variables. But the problem at hand here is, of course, when you combine data from different platforms, it's not that easy to perform statistical analysis across all platforms. So this is not anything that we can recommend doing with our software, because it will require more normalization, just as the requester suggests here. So that's why we focused on the correlation view, because there we just look at the expression profiles over all the samples, and we don't really worry so much about the different normalizations, that protocols that have been used for the different data types. And then another question here: is it possible to integrate all kind of transcriptomic and proteomic data set into the software? Yes, absolutely. So let's have a look at the file again. So as long as you have analyzed data for the same samples, you can merge it into one data set, as we did here, and look for correlation. And then another question, and we have comment: I was expecting to see more multivariate multi-omics integration methods such as CCII or others. Yeah, and unfortunately we don't support that. Another question: how to combine expression data with methylation data? For each gene expression there may be more than one methylation data entry. Yeah, so you must pick which methylation result you want to use, basically. So either you can, in the software, you can actually collapse data to create a mean value, a median, the minimum, the maximum value, if they are, or you manually pick which result you want to use. Perfect. So now back to the request that to show the first the file that we used. To here it is. So here we have, I'll just close that for the moment. So here we have a file with, initially at the top we have genes, and going down we have proteins, and we just merged the data. So we have one gene value and one protein result for each, no, not one, but gene values and protein values for each sample, like this. And again, there might be differences in how these different data types have been normalized and treated on their different platforms. So it's not advisable to do other types of analysis on the just do statistical analysis on this, but working with correlation, that works fine. So any more questions? If not, I just want to remind you about upcoming webinars here. So on Tuesday, this is a bit like an introduction where we look at some simple workflows for doing different statistical tests and finding discriminating variables. And then later on we have an exploration webinar, and further on in February a new basic hands-on training. So we continue to run webinars on different themes. To register or just go in and have a look here at the webinar page. So this webinar has been recorded. Hope that worked. So it will be published on our webpage. So have a look there in a couple of days and you will hopefully find it. So it will be in the videos section of our webpage. Okay, if there are no more questions, you are very welcome to contact me or any of my colleagues if you're already in contact with them. So don't hesitate to reach out to us with any questions you might have. If you haven't already, was that in the previous picture maybe? If you haven't already tested the software, if you're not already a user, you can download a trial license from Qlucore.com, 10-day free trial. So I would like to thank you so much for your attention today. I will send out the slides after this, right after this session. So by that, I wish you a good rest of the day and